Nipah virus is a highly fatal emerging infectious disease found in Southeast Asia. It is classified as a Nipah of the family Paramyxoviridae. The disease was first identified in Malaysia in 1998, but outbreaks have since occurred throughout Southeast Asia. The natural host of the virus are known to be fruit bats of the Pteropus genus. The disease has also been shown to be in an intermediate host in pigs as well. Human transmission of the disease has occurred by direct contact with infected bats, infected pigs, and human-to-human contact. The disease has also been linked to consumption of raw date palm sap contaminated by infected bat excretions. The following slide shows the species of bats known to be carriers of the disease, as well as the geographic location that these bats were found. The following map depicts the geographic location of outbreaks of the Nipah virus. The blue diamonds designate the Nipah virus outbreak, while the red circle demonstrates Hendra virus outbreaks. The blue line depicts the geographic range of the flying fruit bat species Pteropus. The disease is categorized by fatal encephalitis, and symptoms include fever, headache, drowsiness, disorientation, mental confusion, and can progress to a coma within 24 to 48 hours. Respiratory symptoms have also been shown in the disease. Currently, there is no known treatment for the disease. The following slide shows the cases of Nipah virus by location. There has only been 280 known cases of the virus since its discovery in 1998. Unfortunately, the disease has a high case fatality rate of 75%, with 211 deaths occurring since its first being discovered. Disease outbreak of the Nipah virus follows seasonal trends, with the majority of cases occurring between January and May. This seasonal trend has been shown to be a factor in Bangladesh, as the majority of harvesting of raw date palm sap occurs in winter months, most often at night. The first step in identifying an outbreak is to plan for the investigation, and this will require developing an investigation team which will consist of local villagers, local healthcare workers, epidemiologists, local public health networks, as well as laboratory employees needed to confirm and verify the diagnosis, which is the second step. Based on the high fatality of the disease and the low prevalence, a single case is all that's needed to confirm an outbreak. The disease has an incubation period of 5 to 14 days, and once an individual becomes symptomatic, death can occur rapidly. Therefore, a broad case definition is required initially based on clinical symptoms, as well as risk factors, including those who harvest raw date palm sap. Confirmation of the disease can occur through lab work, including throat and nasal swabs, urine, blood samples, as well as cerebrospinal fluid. Based on the low prevalence of the disorder, a cohort study is likely to be used as the principal study design for research into the disorder. A questionnaire will be used to identify the demographics and clinical symptoms of the individuals infected, as well as the risk factors associated with the disease. Questions regarding treatment will be utilized. However, there is no known treatment for the disorder. Data will be tabulated using Microsoft Excel, being categorized by person, time, and place of infection. An environmental investigation will begin and will include harvesting and testing of raw date palm sap to test for the infection, as well as methods to recruit bat species to test for the disease. Despite little being known about the disease, treatment has been focused on antiviral medications which have shown some promise, as well as creating bamboo barriers to limit bat contact with raw date palm sap. Education of the local villagers for transmission of the disease will be an important strategy to help reduce the amount of cases. The differences in outbreaks in Bangladesh and Malaysia raise some questions regarding the route of transmission, as transmission in Malaysia was shown to be occurred by an intermediate host of pigs, while outbreaks in Bangladesh were categorized by direct transmission from infected bats. The epidemiological differences between these two regions will require further research and hypothesis testing to determine the differences. Additional studies are needed to find causation differences between the two regions. Finally, as with any outbreak, communication is vital. This communication will include government officials down to local villagers and is crucial to reducing morbidity and mortality as the disease is classified by a high case fatality.